mechanism to make it in slide form?
我已经成片。Sorry, Professor, I didn't get you.
There are other ways by which you could make it in slide form besides clicking the icon. If your icon does not work below, clicking the icon below does not work, you have to use the icon above.
Yes, Professor.
Vincent,
Yeah.
try pressing F5 in the keyboard.
Yeah. In my computer, I've already make it full screen, but I don't know why the reason in this when I'm sharing the screen, but it is only just like this.
But it's not in full screen. Okay, you try again, Vincent. Is Pax around? Pax, are you around? Pax, are you around?
Does it work right now?
Okay, that's good. That's good, Vincent. It's clear. Loud and clear.
Okay, thank you, Professor. Let us continue the presentation. The topic today is the role of sugar, protein, and fat in diabetes. First of all, I want to discuss the discovery of diabetes. The first use of the term diabetes dates back to 250 BC. The first record of diabetes in medical texts is from 1425. The full name of diabetes is diabetes mellitus, which is composed of the Greek word diabetes and the Latin word mellitus. In 1776, Matthew Dobson demonstrated the sweetness of urine in diabetics and inferred that it might contain a brown sugar-like substance. This is the first time that a clear distinction has been made between type 1 and type 2 diabetes. And then, what is diabetes? Diabetes mellitus is a metabolic disease characterized by hyperglycemia due to defective insulin secretion or impaired insulin action. And the role of sugar in the development of diabetes. When you hear about diabetes, many people think that it is directly related to the sugar in everyday life. But in fact, this is not the same as sugar. Diabetes is a group of chronic metabolic diseases characterized by chronically elevated blood sugar levels. The blood sugar refers to the sugar in the blood, which is directly related to sugar in daily life, but there is no direct relationship between how much sugar is eaten and the diabetes. The root cause of diabetes is insufficient insulin secretion and insulin resistance, which cannot exert its biological efforts, causing a series of diseases caused by tissue problems, not enough insulin or no insulin with considerable biological effect to consume or metabolize sugar. If insulin secretion is normal and can give full play to its biological effect, all the sugar eaten will be broken down. Patients who have diabetes are not unable to eat sugar, are mainly the restriction of total calories intake, such as glucose, fat, and the balance of protein, in order to prevent diabetes from the diet and also to strengthen exercise. Diabetic patients should strictly limit the intake of white sugar, brown sugar, and a series of simple sugars, because these will be absorbed immediately after eating, causing a rapid rise in blood sugar, so that the pancreatic islets at a time are overwhelmed, making the disease aggravated. Number two, the role of protein in the development of diabetes. Firstly, characteristics of protein metabolism in diabetic patients. Number one, inhibition of insulin resistance can cause limited protein synthesis in the body of diabetic patients. Pereira et al. Performed a precise analysis of protein metabolism in the body of type 2 diabetic patients by carbon dioxide labeling and found that insulin resistance inhibits protein synthesis in diabetic patients. Experts believe that since protein is the main functional factor in the body, many of the complications associated with insulin resistance may be related to poor synthesis of certain proteins. Number two, sugar xenobiotic effect. Gluconeogenesis is the process by which the body converts non-sugar substance, for example, sugar-producing amino acids, fatty acids, etc., into sugars, mainly to maintain constant blood glucose and to meet tissue needs for glucose. In other words, even if no sugar is consumed at all, the body will still convert other nutrients into sugar through the gluconeogenic pathway. For example, a large proportion of amino acids, which are the building blocks of proteins, are glycogenic amino acids and can be converted into sugar in the body through this action by enzymes. The glucose clamp test have shown that the rate of gluconeogenesis is significantly higher in diabetic patients compared to non-diabetic patients, and the mechanism is not clear. In clinical work, we often find that some patients mechanically carry out the doctor's orders and diet excessively, interpreting dietary control as strict control of sugar intake, not knowing that other nutrients can also be converted into sugar through gluconeogenesis, such as excessive intake of protein, which can also cause postprandial blood sugar to rise. In a study of starvation therapy conducted by Daska et al. At the University of Prague, Czech Republic, 10 patients with type 2 diabetes and age, sex, and weight matched obese individuals were included as controls, and all patients were examined for glycemia and endocrine metabolism after 60 hours of fasting, and the results showed that hypoglycemia persisted in diabetic patients and that oxidative protein consumption increased compared to controls. Number two, how does protein affect immune function in diabetics? Protein is the material basis of all life activities, and since immunity is part of the physiological functions of the human body, protein is also an important material basis for maintaining the immune function of the human body, in addition to participating in the composition of immune cells. The synthesis of antibodies secreted by lymphocytes, antimicrobial peptides secreted by plant cells, and immunologically active substances such as various cytokines, immunoglobulins, Lysozymes and the complement are also inseparable from proteins. The risk of all-cause mortality is approximately 70% higher in patients with type 2 diabetes with malnutrition than in those without malnutrition. Inadequate protein intake due to overly strict dietary control in some diabetic patients, inadequate protein uptake and utilization by cells due to insufficient insulin secretion or reduced sensitivity in diabetic patients, increased protein consumption due to enhanced gluconeogenesis and long-term protein loss due to proteinuria in diabetic nephropathy, or contribute to protein malnutrition and result in poor immune function. Therefore, diabetic patients should ensure the intake of protein, especially high-quality protein, in their diet to maintain their immune function and enhance their ability to fight infections. Number 3, how diabetics should choose protein-based foods. In addition to animal protein, plant protein is also the main source of protein in the traditional Chinese diet, especially beans and their products. Beans are divided into large beans, including soybeans, black beans, green beans, and mixed beans, including peas, broad peas, green peas, kidney peas, red beans, etc., whose protein content is 75 to 40% and 20% respectively, equal to or even higher than animal food. Soybeans and their products are a good food source of high-quality protein, and the fat is unsaturated fatty acids, which does not contain cholesterol and is rich in bean gum and other soluble dietary fiber. Mixed beans, which are rich in lysines and deficient in methionine, can play a complementary role to protein and increase its protein utilization rate when mixed with methionine-rich cereals. The dietary guidelines for diabetes in China 2017 recommends that staple food are rationed coarse and fine, with whole grains and mixed beans accounting for 1/3. So it is recommended that diabetic patients consume green beans, red beans, and other mixed beans mixed with cereals after processing for the best. Number 3, the role of fat in the development of diabetes. In recent years, the incidence of diabetes has gradually increased and has become a global public health problem. The relationship between pancreatic fat deposition and the occurrence and the development of type 2 diabetes has also received increasing attention. Studies have shown that glucose itself is not responsible for the complications of diabetes, especially in patients with type 2 diabetes. Rather, responsibility lies with a range of factors, including obesity, hypertension, dyslipidemia, inflammation, and insulin resistance that impact on adipose tissue fatty acid metabolism, which underlies the development and the progression of diabetic nephropathy, retinopathy, and neuropathy. Dyslipidemia is a major factor in the pathogenesis of diabetic neuropathy, and increased plasma triglycerides are associated with progression of neuropathy in type 2 diabetes, while obesity and systemic dyslipidemia are independently associated with an increased risk of developing neuropathy. Lipid-lowering therapy is associated with a reduced risk of lower extremity amputation, suggesting that lipid lowering may be beneficial for diabetic neuropathy. Fat distribution in the pancreas and its pathological role. Cellular fat can be divided into intracellular fat and extracellular fat. Intracellular fat refers to triglycerides distributed in islet and alveolar cells. Extracellular fat refers to adipocytes or adipose tissue distributed in the interlobular interval of the pancreas or replacing the pancreatic parenchyma, which can also be divided into interlobular fat and intralobular fat according to anatomical location. Intra-islet cell triglyceride distribution is mainly related to leptin and insulin action. In obese people, relatively low leptin or defective leptin action inhibits diminished lipid synthesis and enhanced intracellular esterification in pancreatic islet, leading to triglyceride distribution in islet cells. High insulin and hyperglycemia can affect lipid synthesis, leading to triglyceride distribution in islet cells, and hyperglycemia leads to reduced mitochondrial beta oxidation, allowing triglycerides to accumulate intracellularly. Insulin resistance promotes peripheral lipolysis, increased free fatty acids in the circulation, and prolonged exposure of islet beta cells to high glucose and higher levels of free fatty acids lead to increased intracellular triglycerides. Number two, correlation between pancreatic fat deposition and type 2 diabetes. Pancreatic fat deposition is correlated with some pancreatic diseases or metabolic syndromes and its etiology leading to pancreatic alveolar necrosis may lead to fat replacement of normal pancreatic tissues. Cystic fibrosis or mucus viscosis of the pancreas, acute or chronic pancreatitis, and pancreatic cancer may lead to replacement of pancreatic tissues by fat tissues. And metabolic syndromes such as obesity, non-alcoholic fatty liver, and type 2 diabetes are associated with pancreatic fat deposition. Pancreatic fat deposition is closely related to the development of type 2 diabetes and beta cell dysfunction. Quicklet et al. Found that obese New Zealand mice fed with a high-fat diet had increased pancreatic adiposity, impaired islet cell function, and increased blood glucose level. Pancreatic adipocytes induce excessive insulin secretion and release higher levels of free fatty acids compared to adipocytes in white inguinal adipose tissue, while insulin resistance is also a causative factor of fatty liver. Pancreatic steatosis causes beta cell dysfunction through the interaction of adipocytes with pancreatic islet cells. Compared with fatty liver, fatty pancreas is a stronger risk factor for type 2 diabetes. Animal experiments confirmed that young diabetic models rats receiving a high-fat diet for a long time were the first to develop insulin resistance. followed by impaired pancreatic beta cell function, elevated blood glucose, and diabetic signs after 8 to 16 weeks of feeding, and their diabetes may be related to triglyceride deposition in the pancreas due to a high-fat diet. Wang et al. Conducted a controlled study of subjects with and without non-alcoholic fatty pancreas and found that the incidence of diabetes was higher in the former than in the latter, and that non-alcoholic fatty pancreas was significantly associated with the development of diabetes. Lu et al. Measured the fat content in the pancreas of diabetic patients by MRI three-point Dixon colon and confirmed that the fat content in the pancreas was higher in diabetic patients than in normal subjects. All of the above findings suggest that pancreatic adiposity is closely associated with type 2 diabetes. In addition, intermittent fasting may reduce the pancreatic fat content and prevent the development of diabetes. Animal experiments revealed that the blood sugar level of diabetic model rats fed with a high-fat diet increases significantly at about 10 weeks to become a diabetic rat, while their pancreatic fat content started to increase at about 4 weeks and increased significantly at 8 weeks of age, indicating that the pancreatic fat content of rats started to increase before the onset of type 2 diabetes. Maggio et al. Found that pancreatic fat content was higher in obese than non-obese people, and impaired pancreatic beta cell function was seen in obese people without diabetes, further suggesting that increased pancreatic fat content precedes the onset of type 2 diabetes. Hanskison et al. Found that pancreatic fat content with circumference, subcutaneous, and visceral fat were reduced in patients with pre-diabetes and type 2 diabetes through exercise training interventions. The pancreatic fat infiltration was reduced and the pancreatic beta cell function was improved. Gabbard et al. Found that patients with reduced pancreatic fat content and improved insulin resistance after restricting caloric intake through bariatric surgery, pointing out that reduced pancreatic fat content may help to improve islet beta cell function. It can be seen that pancreatic fat deposition is closely related to the occurrence and development of type 2 diabetes, and the monitoring of pancreatic fat content can enable early detection and diagnosis of pre-diabetes and timely intervention. Then it's the complications in diabetic patients. Diabetes itself is not terrible. What is terrible is the harm caused by its complications. Diabetic patients are often combined with cerebrovascular pathology, which can lead to cerebral hemorrhage, stroke, subarachnoid hemorrhage, lacuna, cerebral infarction, cerebral embolism, and cerebral thrombosis. Strict control of blood sugar, blood pressure, blood lipids, weight, and other comprehensive treatment is an important measure to improve the cardiovascular and the cerebrovascular pathology. Untreated diabetes may cause very dangerous acute complications such as diabetic ketoacidosis, diabetic non-ketotic hyperosmolar coma, etc. Finally, how to prevent diabetes in your life. Number one, aerobic exercise. For example, cycling, jogging, and other aerobic exercises that can enhance physical fitness and effectively prevent type 2 diabetes. Number two, eat more fruits and vegetables. Fruits and vegetables have many vitamins, which are very important for health and help lower blood sugar in the body. Number three, avoid eating a lot of sugary food and drinks in your daily life. Control drinking and sugary food. Drink more plain water. Number four, eat more foods containing omega-3 fatty acids. Can be effective in preventing diabetes, such as eating more fish foods. Number five, regular life. Go to bed early, get up early, and don't eat too much. Smoke and drink as little as possible in your daily life. Then it's the summary. Number one, there is no direct relationship between the amount of sugar eaten and diabetes. The principle of diet for diabetic patients is to have a balance of energy in and out. And even if they eat sweets, they can metabolize the the sugar with increased exercise. However, diabetic patients should try to eat less sweets because the sugar inside these sweets is monosaccharide, which is easily absorbed and raised blood sugar faster. Number two, protein is one of the three major energy supply substance in the body and has little effect on diabetes and has no therapeutic effect. Protein is also a caloric food, and protein can be converted into glucose through gluconeogenesis. So diabetic patients should control the amount of protein intake and generally require that the protein content is the diet does not exceed 15% of the total calories. So moderate intake of protein is useful for the body, but not in large quantities. Number three, there is a strong correlation between obesity and diabetes, and obesity increases the risk of diabetes. Adipose tissue is the source of the development of diabetes. That will be the end of this speech. Thank you for your listening.
Okay, Vincent,
Yes.
thank you for the very informative. You just put first your slide. Just put your slide in the screen. Vincent, you just put your slide on the screen.
Okay.
We have to. I have to make some clarifications regarding your report.
Okay, which one?
Okay, just put it there, and then make it slides. Okay. Perhaps you could start the first slide or the second slide. Okay. What Vincent has been presenting is about the effect of first carbohydrate, second protein, and third fats on the development of diabetes mellitus. He tried to explain the correlation between sugar and diabetes. Why, if you take a lot of food containing sugar, the possibility of developing diabetes is very high. And also, Vincent also mentioned about the possibility of developing diabetes if you keep on eating protein. In other words, it does not only diabetes. The disease does not only concern carbohydrate, but rather it also concerns protein. And he tried to explain why eating protein can cause diabetes mellitus. Okay. And it has something to do with the amino acids that are considered as glucogenic amino acids. You mentioned that about glucogenic amino acids. What does Vincent mean is that this glucogenic amino acid can be converted to glucose by way of gluconeogenesis. By way of gluconeogenesis. Okay? And then another thing he also explained about fats, that fatty acid triglycerides could also cause diabetes mellitus. The intake of fats, fatty foods, especially foods that are unsaturated fatty acid, that contains unsaturated fatty acid, it has high possibility that it can cause diabetes, that a person will develop diabetes. And the primary reason is the accumulation of fats in the islets of Langerhans, in the pancreas. And when these fats, triglyceride, accumulates in the islets of Langerhans, the first thing that will be affected are the beta cells. Are the beta cells in the islets of Langerhans. And you know what is the function of the beta cells? Vincent, do you know the function of the beta cells in the islets of Langerhans?
Sorry, I don't know.
You do not know?
Yeah.
Okay, who reported on that? Somebody has reported on that. Anyone from the class?
Hello, Doc. Beta
Okay,
cells po,
Michaela.
beta cells secretes the insulin po.
What? Again?
Beta cells secretes insulin, Doc.
Vincent, beta cells in the islets of Langerhans, Sora, beta cells in the islets of Langerhans is responsible for releasing or secreting insulin. That is why, okay, it is responsible. That is why if the beta cells is impaired or the beta cells are non-functional because of the presence of fatty acid, because of the accumulation of triglycerides along the surface of the islets of Langerhans, affecting the beta cells, insulin could not be released. Or in other words, the release of insulin will be affected. It will be hampered. And so what will happen if insulin is not released from the beta cells of the islets of Langerhans? The glucose in the blood will accumulate. And the primary reason is because the glucose could not be utilized by the cell. The glucose, in other words, could not enter the cell, and then it will be utilized, it will be metabolized to undergo the process of glycolysis. That is the primary reason. Why? Oh, this is the primary reason why obesity is related. It is being related or being it is related to the development of diabetes mellitus. Because of that. Because of that. Okay. And you have mentioned, Vincent, a lot of studies that were done to show that diabetes mellitus could be also caused by the accumulation of fatty acid based on the different experiments that were conducted by many researchers. By many researchers. And what is bad about having diabetes is because of its harmful effects on the internal organs. Internal organs, like for example, we could say primary organs of our body, like for example, the kidney, the liver, the heart, and also the nervous system. One is there is what is known as nephropathy. It can cause nephropathy, meaning that the kidney will be impaired because the nephrons in the kidney will be destroyed. And you know, the nephrons, these nephrons, they are the one responsible for the filtration of the urine. Filtration, it is a filtration system of the kidney. And once that nephron cells are destroyed, the filtration system of the kidney will be affected, and then there is what is known as kidney failure. And then it will also affect the heart. It will also affect the heart. Diabetes also will affect the heart. And you know, as you have mentioned, that the heart is affected if there's an increase in glucose because of the blocking effect, because of its blocking effect, because of the formation of fats in the blood vessels that supply the heart. There is also atrophy of the heart, especially when the heart is blocked. That means that the supply of oxygen in the heart will be affected, and some of the cells, cardio cells, some of the cells in the heart will be affected. Okay? Neuropathy. This is the effect on the nerve cells, neurons. There will be also degeneration of the nerve cells because the nerve cells degenerate because also of the increase in blood sugar level. Blood sugar, sir, it will affect the nerve cells. But how about protein? Why is it that when there is a large amount intake of protein, again, there will be an increase in the blood sugar level. In the blood sugar level. Can anybody explain that? Why, if there is an increase or uptake of protein by our body, there is a high probability that diabetes, diabetes will occur. Okay? Can anybody explain that? Michaela? Can anyone? Jaira, are you in? And Angel, are you in? Angel? Angel absent?
Doc, yes, Doc.
Yes. Angel, do you know why? If there is an increase in the uptake of protein in our body, there is a possibility that you will develop diabetes mellitus. Ah,
Doc,
Angel?
for a high for high protein diet, it lowers the blood glucose postprandially in persons with type 2 diabetes, and it also improves the overall glucose control po.
I am talking about when there is a higher uptake of protein, you will develop, a person will also develop, there will be an increase in blood sugar level. Why? Are you listening to Vincent? Are you listening to Vincent report that there is, if there is a higher uptake of protein, the person will develop also the blood sugar level will also increase. Huh?
Yes, Doc. Opo, yes, Doc.
Oh, why? How come? How come, Angel? How about you, Alexander? Alexander, are you in?
Yes, Doc, I'm in po.
Okay.
A high intake of protein uh, will contribute to diabetes because of gluconeogenesis pathway, wherein protein can be turned into glucose.
What will be turned into glucose? What is specific?
Ah,
Specifically, what will be what will be converted to glucose?
what from the protein is made up of amino acids. These amino acids will be converted into pyruvate, and then uh, phosphoenol pyruvate, and then
Hindi
glycerol.
pa.
Ay,
Paano kagad yun? Why? Magiging pyruvate. Pyruvate magiging phosphoenol pyruvate. Magiging phosphoenol pyruvate ba kagad yan? Will it be phosphoenol pyruvate
no, not
immediately?
not yet, wala, Doc, not yet po.
Oh,
So
so
pyruvate inside the mitochondria will first be converted into oxaloacetate, and then oxaloacetate
prior to that, what will happen to pyruvate?
uh, pyruvate is ano po yun? Pyruvate is carboxylated po. By pyruvate
Will it be
carboxylate.
converted immediately to oxaloacetate? Will be pyruvate? Will pyruvate be converted immediately to oxaloacetate
Ah,
in the mitochondria? Anyone who can help Alexander? Pax, are you in? Pax. Pax is absent. Joel, are you present, Joel?
Yes, Doc.
Okay. Is pyruvate immediately converted to oxaloacetate in the mitochondria, Joel? You don't know? Joel?
Doc,
Yeah.
pyruvate is converted to acetyl coenzyme.
Acetyl coenzyme. What? Acetyl coenzyme what? Oh, Joel, you do not know acetyl coenzyme what? Raquel, are you in? Raquel. Raquel? Absent? Earl. Kirsten.
Yes, Doc.
Okay. Earl, Kirsten, what will happen to pyruvate?
Um,
In,
Pyruvate will uh, be oxidized into acetyl coenzyme A,
oh,
and by that,
have you heard that? Have you heard that, Joel? Acetyl coenzyme A. Hindi lang acetyl coenzyme. Acetyl coenzyme A. Okay? Kirsten,
yes, Doc, and then this acetyl coenzyme A will bind to oxaloacetate, and it will become citric acid,
will what? Will what?
and will become
It will,
citric acid.
it will what?
It will merge or somehow act with oxaloacetate, Doc, the acetyl
What is
coenzyme.
the term? What is the term? What is the right term, Earl?
um, Merge or react. Tama ba yun, Doc? With oxaloacetate.
Condense.
Yes, yes. And then it will become citric acid. And it will undergo na, Doc, the citric acid cycle.
Okay? To form again what?
Oxaloacetate again, Doc.
And then, what will happen to oxaloacetate?
And then oxaloacetate, ano nga yun?
It will undergo gluconeogenesis to
Opo.
form what?
Pyruvate again, Doc.
Magiging pyruvate siya? Magiging pyruvate ulit ang oxaloacetate. Anybody, anyone? What will happen to oxaloacetate in the mitochondria? What will happen?
Doc?
It will
Yes,
phosphoate.
Alexander. Yes, Oscar. talking that the that the protein, the amino acids of the protein, like for example, for example, phenylalanine, lysine, methionine will be converted to pyruvate, will be converted to malate, will be converted to oxaloacetate. These are the compounds found in citric acid cycle. And then eventually, this will be converted to oxaloacetate, which is the precursor in the synthesis of glucose. Okay? Okay, Vincent? Are you
Yes.
still in? Okay, that is how you explain this, how you present this topic. Okay? How you presented this topic. The important role of carbohydrate, protein, and fats in the development of diabetes mellitus. Diabetes mellitus. Are you diabetic, Vincent? Vincent, are you diabetic?
No, I'm very healthy.
Are you healthy? And you are sports-minded? Are you sports-minded? You have been talking here of exercising in order that utilize your blood sugar. Are you
Yes.
fond of exercising, Vincent?
I do love playing basketball.
Basketball. Okay. That is a form of sports, form of exercise. How about Sura? Sura, are you in? Sura, are you in?
Yes.
Okay. Are you diabetic, Sura?
No.
No. Okay. Are you also fond of sports? Playing sports?
我看
你喜欢做运动吗？你喜欢做运动吗？
yes.
What kind of sports, Sura?
你喜欢做
Are you in?
She told me that she likes jogging.
Jogging. Okay. That's good. That is really. I think obese person should always jog. Jogging, not really jogging, but light, but walking every day. Okay, Vincent. Thank you.
Yes. Thank
Okay.
you, Professor.
Okay. Who is the next reporter? By the way, Pax is not around. The one who is that guy who is supposed to report last Saturday but did not report? Are you Oscar?
Yes po, Doc.
Okay. You start
笑
now.
死我。
Oscar, start
Yes.
now.
Okay po, Doc. For a while
What
po.
is your report, Oscar?
It's about metabolic regulation, anabolic and catabolic regulation of carbohydrates.
Okay. That's good. Vincent?
Yes, Professor.
Sora will have a second report. Okay,
Second.
a second. I think I think the topic, I think it was posted. The topic was posted in the Google Classroom.
Okay, I see.
You kindly check the Google Classroom
Thank you, Professor. I know.
Oh, okay. Okay, Oscar?
Can I start po, Doc?
Yes, you start.
So, good afternoon, everyone. So, for today's discussion, we'll be talking about metabolism of carbohydrates. So, we'll be focusing on the anabolic and catabolic regulation. So, at the end of this discussion, we will be able to understand the basics and principles of metabolic regulation and identify the key factors in regulation of glycolysis and gluconeogenesis. So, to start, as definition, metabolism is the sum total of all biochemical reaction that takes place in a living organism. So this can be classified into two, catabolism and anabolism. Catabolism pertains to metabolic reactions in which large biochemical molecules are broken down into smaller ones, which results to the release of energy. On the other hand, anabolism includes metabolic reactions in which small biochemical molecules are joined together to form larger ones, which requires the intake of an energy. Thus, this series of chemical reactions forms a sequence in which they can take form in a linear or cyclic form, and we call them as metabolic pathways. With the discovery of these metabolic pathways, scientists were able to understand how energy is being stored and produced, and also we were able to identify the process of synthesis of the building blocks of other biological substances or organic substances. And also, with these pathways, we were able to identify or know how do we eliminate waste materials inside our body. So, it is very important that the normal process of this metabolic pathways take place, that no step should be defective. Once there is a defect in any of the steps of these pathways, then that is a major contributory factor to the development of human disease. So, for each metabolic reaction in a pathway, the substrate is provided by preceding reaction at the same rate at which it is converted into product. Thus, although the rate of metabolite flow, or what we call flux, so through this step of the pathway may be high or variable, and the concentration of the substrate remains to be constant. So for the two-step reaction where V1, or the first rate of metabolite flow is equals to the second rate of metabolite flow, so the substrate concentration is constant. And with this, the homeostasis of our body, or the chemical equilibrium of the biochemical substances in our body, must be maintained and regulated. So now we identify the factors which might affect the rate of biochemical reaction. The first one is the concentration of reactants and the products. The second one is the activity of the catalyst, which depends on the concentration of the enzyme, and as well as the intrinsic activity of the enzyme. On the third one, concentration of effectors also affects the biochemical reaction, which depends on the allosteric regulators and competing substrates such as the competitive inhibitors and non-competitive inhibitors, and as well as factors such as pH and ionic environment could affect the biochemical reaction. And lastly, although it is not usually associated with, or not usually affects the rate of reaction, still temperature is a factor need to be considered, because it can still affect the rate of the biochemical reaction. So in the biochemical reaction in our body, one particular biochemical substance we call enzyme plays the key role. So for this substance to be synthesized, we should be able to understand how, for us to be able to understand the role of this particular biochemical substance, we must learn how thus enzymes are being synthesized. So to start, extracellular signals such as hormones, growth factors, and cytokines attaches to the cell's receptors or penetrates the cell membrane, further activating what we call the transcription factor. This transcription factor now will bind to a specific target in our DNA we call the response element. Now, this response element will be our target DNA. This target DNA will code for the target gene that codes for a specific enzyme that we wanted to produce. So upon detection or upon setting the initial or setting the starting point of the transcription, mRNA could be now transcribed. So upon the transcription of the mRNA, for it to become an enzyme, it needs to be translated in the ribosomes. So mRNA inside the nucleus needs to go outside the nucleus, going through the ribosomes. Before reaching the ribosomes, the right amount...
Oscar, mRNA is not transcribed.
The DNA po is
what is transcribed is the DNA into mRNA. What is being shown in there is the mRNA transcript. Huh?
Noted po.
In the nucleus, there is the DNA and there is the mRNA transcript. It is the DNA that is transcribed. You repeat your explanation.
Okay. So as the transcription factor binds to the response element or the target DNA, the DNA fragment that was targeted will be transcribed into mRNA inside the nucleus. For this mRNA to become an enzyme, mRNA needs to be translated outside the nucleus and needs to go to the ribosomes. So mRNA goes out of the nucleus, and then the right amount of mRNA also must be maintained for regulation. Excess mRNA products would be degraded by ribonucleases. Upon achieving the optimum number of mRNA needed for the protein synthesis, the mRNA will be translated into the ribosomes. So the mRNA products now, as translated in the ribosomes, will become a series of amino acid chains. This amino acid chains will be folded and become a protein and later on become an enzyme. So this enzyme now is the main key regulator of the biochemical reactions inside our body. So enzymes can undergo these 6 different regulatory mechanisms. Although enzymes have a finite lifespan, which can vary from hours to days. So the enzymes that are attached with ubiquitin will undergo protein degradation in the proteasome. While enzymes in the subcellular organelles would undergo sequestration, particularly in the endoplasmic reticulum. Some of the enzymes could also bind to substrates, others could bind to ligands, and few would undergo, some would undergo phosphorylation or dephosphorylation, and some will be inactivated when enzyme combined with the regulatory protein. So to further understand the phosphorylation and the phosphorylation of enzymes, so here we can see that phosphorylation and dephosphorylation happens in the hydroxyl groups. So phosphorylation is catalyzed by enzymes we call protein kinases, whereas the hydroxyl group will gain a phosphate group from ATP with the action of protein kinase. This ATP will become ADP thus donating or giving off the 1-phosphate ion to the hydroxyl group. On the other hand, dephosphorylation can happen with the presence or through the action of phosphoprotein phosphatase. With the hydroxyl group with the phosphate ion, it will lose the phosphate ion, thus leaving an hydroxyl group without a phosphate group. Thus, dephosphorylation can happen or catalyze via the presence of an enzyme, or it can just be done spontaneously. One factors that I have mentioned a while ago is the rate of the substrate. The rate of the substrate could affect the rate of the biochemical reaction. So in here, we can see on this plot diagram that the rate is more sensitive to low concentrations. So we can see here that ATP is the substrate, and here we have the initial velocity. We can see here that in low concentrations of the substrate, there is a high rate of reaction. However, as the levels of substrates increases, the levels of the rate of reaction also decreases or maintains a stagnant or plateau phase. So on this particular slide, we can understand that the concentration of the substrate could affect the rate of the biochemical reaction. Next one, as I have mentioned that ATP is used as substrate, so when ATP is consumed and such that its concentration drops at least 10%, the relative increase in AMP is much greater than that of ADP. Thus, we have now the AMP activated protein kinase, which would respond to an increase in the AMP by phosphorylating key proteins and thus regulating their activities. And the rise of AMP may be caused by reduced nutrient supply or by increase in exercise. So we can see on the table here that this was the concentration before the reaction took place. So we have here ATP, which reduced at least 10%. Okay? So we can say that based from this statement, that relative drop of 10% would have a greater increase in AMP, thus showing that when ATP have a relative change of 10%, AMP had a 600% increase in the change. So let us identify the role of AMP-activated protein kinase in the carbohydrate and fat metabolism. So as I have said a while ago, when ATP levels are depleted, okay, AMP levels would be increased. So with the increase of AMP levels, this would trigger the activation of the AMPK. Aside from the increased levels of AMP, such factors such as exercise and the release of leptin and adiponectin, which are responsible for appetite, would also trigger the activation of the AMPK. This AMPK, okay, will signal or will activate the brain and will signal our body to increase the food intake because of reduced nutrient supply, in which that would lead to the degradation or depletion of energy source. While there is also an active fatty acid update or fatty acid oxidation in the skeletal muscles and on the heart. While the fatty acid synthesis on the adipose tissues and the liver would be inhibited, as well as the secretion of insulin on the pancreatic beta cell.
in multiplex. So, di ba sa multiplex, pwede kayong maramihan.
So, some of the enzymes in the pathways limits the flux of metabolites more than the others. Usually, these enzymes are the ones we call regulated. So, these regulated enzymes have not reached the equilibrium yet. And not all regulated enzymes have the same effect on the entire pathway, in which some regulated enzymes control flux and some regulates the steady state concentration of metabolites.
Primers, polymerase, RT-PCR, or
Among these enzymes, two known enzymes are the hexokinase and the phosphofructose kinase. So these are appropriate targets for regulation of the glycolytic flux, which is responsible or which is associated with glycolysis. An increase in levels of hexokinase activity would enable activation of glucose. Thus, an increased levels of fructokinase 1 activity enables catabolism of glucose via glycolysis. So we can see here now that it was proven that the rate or the concentration of the enzyme have affected the rate of reaction. Thus, we can see here the enzyme phosphofructokinase and the enzyme hexokinase with increasing enzyme concentration showed an increasing enzymatic activity. Although it was noted here that hexokinase have exhibited a much greater enzymatic activity. So now we'll talk about the carbohydrate metabolic regulation. On this part, we will be focusing on glycolysis and gluconeogenesis, our model. So in here, in the carbohydrate metabolism of particularly of glucose, glucose needs to be converted into glucose 6-phosphate as the initial action in glycolysis. So for it to be converted into glucose 6-phosphate, glucose needs the action of the enzyme hexokinase. However, it was noted that hexokinase, particularly the hexokinase 1, is expressed in all tissues and to different levels, and these are also being inhibited by the glucose 6-phosphate. Thus, glucose 6-phosphate, which is a product of the conversion of glucose with hexokinase, would inhibit the process of glycolysis. So, an isozyme of hexokinase, which is hexokinase 4, which is only expressed in the liver, is the one that is not inhibited by glucose 6-phosphate, which now serves to clear the blood glucose and stores glucose as glycogen. So, hexokinase 4 is the one that is responsible for continuation of this glycolytic flux. On the second part, we can see here that glucose 6-phosphate would be isomerized into fructose 6-phosphate with the action of the enzyme phosphohexose isomerase. So this now fructose 6-phosphate to be converted into fructose 1,6-bisphosphate is the commitment point, whereas if we have this fructose 1,6-bisphosphate, this would ensure the flow of our glycolytic flux. So we need to ensure that fructose 6-phosphate must be converted into fructose 1,6-bisphosphate via phosphorylation. However, again, in cases or in conditions where ATP is increased, where there is an excess supply of ATP in our body, the conversion of fructose 6-phosphate to fructose 1-bisphosphate will be inhibited. So, the glycolysis would then be not finished or done. But as I have mentioned earlier, with the
The virus
On
don't
the
cause
need
cervical
of the
warts
AMP and the ADP, which are products of this will activate or continue the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. Also, on the allosteric side of the fructose 6-phosphate, citrate from the product of citric acid cycle would inhibit fructose 6-phosphate conversion into fructose 1,6 bisphosphate, thus inhibiting the glycolytic pathway. However, in the presence of a metabolic regulator, which is fructose 2,6 bisphosphate, which is again not to be confused with fructose 1,6 bisphosphate, okay, will activate the fructose 6-phosphate conversion into fructose 1,6 bisphosphate. We must take note that fructose 1,6 bisphosphate is an immediate product of glycolysis, but fructose 2,6 bisphosphate is only a metabolic regulator. So to differentiate these two, this one will continue to undergo the glycolytic pathway, but this one would remain to be here. Now, how did we come up, or how did fructose 2,6 bisphosphate activated the fructose 6-phosphate conversion to fructose 1,6 bisphosphate? So it is through the action of the enzyme PFK2, or the phosphofructokinase 2. So fructose 6-phosphate would be phosphorylated by phosphofructokinase 2, converting into fructose 2,6 bisphosphate, which is again used in the activation or the conversion of fructose 6 bisphosphate into fructose 1,6 bisphosphate. So, in summary, we can see here that the relation of the F26BP, okay, or the fructose 2,6 bisphosphate, will activate PFK1, which is, again, PFK1 will convert fructose 6-phosphate into fructose 1,6 bisphosphate. But the absence of F26BP, or the fructose 2,6 bisphosphate, would activate the fructose biphosphatase 1, which would trigger the phosphorylation of fructose 1,6 biphosphate into fructose 6-phosphate. So in here, we can see the association of the hormones insulin and glucagon. When sugar levels are being increased, insulin would be secreted. So insulin would trigger the activation of PFK2, in which again the phosphofructokinase 2 leads to the activation or conversion of fructose 2,6 bisphosphate. Okay. Which would then, an increased levels of F26BP would stimulate glycolysis and would inhibit gluconeogenesis. On the other hand, when glucagon is secreted with an increased levels of cyclic AMP-dependent protein kinases, this would activate the FBPase2, or the fructose biphosphatase. Okay. This activation of the fructose bisphosphatase rather, would inhibit glycolysis and stimulate gluconeogenesis. So we can see the relation of the phosphofructokinase and the fructose 2,6 bisphosphate in the regulation of the glycolysis. So after the conversion of fructose 6-phosphate into fructose 1,6 bisphosphate, this would be further converted into phosphoenol pyruvate, in which this phosphoenol pyruvate, with the action of pyruvate kinase, would then produce pyruvate, which will now serve as energy. So in here, pyruvate kinase can be activated by fructose 1,6 biphosphate and inhibited by signs of abundant energy. And this can also be activated by phosphorylation in response to signs of glucose depletion. In this diagram, we can see here that, as I have mentioned earlier, that fructose 1,6 bisphosphate rather, activates phosphoenol pyruvate conversion into pyruvate. However, in cases of high levels of ATP and also an increased levels of acetyl coenzyme A, which might be a product of um, um, citric acid cycle, would inhibit the conversion of phosphoenol pyruvate into pyruvate. Also, alanine can also inhibit the conversion of PEP into pyruvate. On the other hand, in the liver, we can see here that glucagon, okay, will now inactivate pyruvate kinase. Thus, the inactivation of pyruvate kinase would then stop the utilization of glucose in the liver or storage of glucose in the liver, thus distributing the glucose into the other parts that needs energy. So we can see also here that there are two alternatives of pyruvate. So this can be a new source of glycogen, can be a stored energy as glycogen. My apologies for the typographical error. And also, it can be used to generate NADPH via pentose phosphate pathway. And also, pyruvate can be a source of acetyl coenzyme A, which can store energy as body fat or make ATP in the citric acid cycle. Also, acetyl coenzyme A stimulates glucose synthesis by activating pyruvate decarboxylase, which is needed in the conversion of pyruvate to oxaloacetate to now enter the process of gluconeogenesis. So, aside from the mentioned enzymes, so we can see here that I would not be focusing on this. These are the genes that are regulated by insulin that causes regulation of the following pathways. So, that would be my reference for this topic would be the Leininger Principles of Biochemistry and Stoker Biochemistry 2, second edition. Thank you very much.
Oscar.
Yes,
Okay,
po,
Oscar.
Doc. Yes po, Doc.
Okay, just give me one slide.
Where po?
About I think the first figure that you have shown.
This one,
Not
Doc.
that one. Not that one.
This one.
Yeah, that one. Okay, make it slides. Slide form. Okay. What is this regulation all about? What is being regulated in here?
On this diagram though, it was described here how enzyme is produced and the regulation of enzyme, regulatory mechanism of enzyme.
Okay. This is the basis for your further explanation on how enzymes are regulated in glycolysis, gluconeogenesis, and other pathways. Okay. This will be your basis. Okay. And you are talking here of extracellular, cellular signals. And then, what is your ligand in here?
The
in your
ligand,
in your figure, okay, wherein there is a receptor. What will be your ligand?
so for the ligand, it depends on what target gene that you would like to produce. So that would depend.
So in this case, what will be is supposed to be your ligand?
On this case, doc, it was not specified what type of enzyme is produced, so it was not stated here.
Your ligand could be hormone in here.
Yes, doc. Hormone or either growth factors or cytokines, doc.
Okay. So what will happen to your ligand?
That would bind to, that would lead to the activation of the transcription factor, doc.
Transcription factor. And how it will be activated? Your
Once
transcription factor.
transcription factor bind to a response element, doc, or upon the binding of the extracellular signals with the transcription factor, that would bind to the DNA fragment or the target DNA po, which would then serve as the template for mRNA transcription, doc.
So you have the extracellular signal
Yes,
and
po.
you have the transcription factor. Okay. And then you have the receptor, you have the ligand. Okay. When the ligand binds into the receptor, what happens?
It will now select the DNA fragment, doc, that is the DNA fragment of the target gene that we want to that codes for the specific enzyme that we wanted to produce.
The first thing that will happen, and then when your extracellular signal will enter, it will enter the cell. Your extracellular signal will enter the cell. There are two ways by which what happen. There are two ligands in there. Okay.
Yes,
There are
doc.
two ligands in there. The one that will bind into the receptor, and the other one is that will enter the cell.
Yes, doc.
Did you see that?
Yes, doc.
Okay. When this ligand bind into the receptor, and then another ligand will enter the cell, so what will happen?
From that, doc, the response element or what we call the target DNA region, the target DNA would be identified as the starting point for mRNA transcription, doc.
For a transcription. So what will happen to the transcription factor and to this extracellular, to this ligand? What will happen to this transcription factor and then this ligand?
For a moment, doc.
You look at your, what will happen to the transcription factor?
It will bind to the DNA fragment, doc.
The transcription factor, before it will bind to the DNA fragment, it will enter the what?
Nucleus, doc.
Huh?
It will enter the nucleus po.
It will enter the nucleus. Okay. And what is the role of that ligand that entered the cell?
It will serve as the it will serve or identify the starting point of the mRNA transcription, doc.
And so it will bind into the transcription factor, and then it will bind into the
DNA fragment.
and what do you call that? where it will bind into the promoter region of the DNA, and then transcription will start.
Yes po, Doc. Promoter region.
Okay, it will start the transcription process to synthesize what?
The protein, Doc, or the enzyme that is
Ano mo masisynthesize dyan? When the DNA is transcribed, what will be synthesized? What will be formed?
mRNA first, Doc.
Ha?
Upon the binding of the transcription factor to the promoter region, so it will serve as a starting point. Then mRNA would be initially synthesized, though.
By transcribing the DNA. The
Yes.
DNA will be transcribed, and then mRNA will be synthesized. And then what will happen to the mRNA?
So mRNA, for it to be translated po, it needs to go outside the cell nucleus and go through the ribosomes, though.
And then bind with the ribosome, and then there will be translation. The mRNA will be translated into protein.
Yes, Doc.
Okay. And you also mentioned that there about mRNA degradation.
Yes.
Okay. If, for example, the mRNA will not be translated, that means that it will be degraded.
Yes, Doc.
MR. And do you know how mRNA could be prevented from degradation? How it could be prevented from degradation, the mRNA?
I'm not sure po, Doc.
You are not sure. We have explained this, how mRNA could be prevented from degradation. Anyone? How this RNA could be prevented from degradation? Anybody? Anyone who could explain how the RNA could be prevented, messenger RNA could be prevented from degradation? Yulimel, are you present? Yulimel. Kenneth, are you present? Kenneth?
Yes, Doc.
Oh, can you help Oscar? How could you prevent the degradation of mRNA?
During synthesis, Doc, it happened until one cup at the to protect mRNA from the
No, you're choppy, Kenneth.
wait, Doc.
How could you prevent the degradation of mRNA? Kenneth?
Yes, Doc. Hello, Doc. Sorry.
Okay.
Current Doc, it is by addition of the 7 metal bonds in cap, which will protect the mRNA from degradation,
And?
and poly-A tail.
Okay, did you understand that, Oscar?
Yes po, Doc. Poly. Yes, Doc.
That actually mRNA, once it is bind into the ribosomal RNA, That means it is protected by polyaethyl cup and metal 7 guanosine cup. That's why that MRA degradation will not happen. Oscar?
Yes, Doc. Noted po.
It will not happen. So it will be translated into protein. Okay, enzyme. Okay? It will be translated into protein. Now, again, in your second diagram, there will be a lot of regulation. Okay, your enzyme. Okay? The
Yes,
first
Doc.
thing that will happen is that what will happen to the enzyme when
When it is
what?
attached to a ubiquitin, Doc, it will undergo protein degradation in the proteasome.
No, now we are talking now of your enzymes, which will be regulated. Okay? Your enzyme, what will happen to the enzyme? It will be regulated on the next on the next on the next diagram. Now,
Yes, one,
no,
Doc.
no, that is not
Yes, one.
okay. Now your messenger RNA is now translated into an enzyme. Huh? Can you see the diagram? Enzyme,
Yes, Doc.
and then there are a lot of regulations.
This one, Doc.
Huh? There are a lot of regulations. Okay, so is the first regulation involved?
Here, Doc, the first one, it's protein degradation.
Is protein degradation, and then and protein degradation is how it will happen, how the protein, how the enzyme will be degraded.
When it is attached to ubiquitin, Doc, it will be degraded in the proteasome.
Okay, so that is one. There will be protein degradation. Then another one is what? The
Enzyme
enzyme,
Here, Doc, enzyme sequestration.
enzyme sequestration in the where? Where will it happen?
And the subcellular organelle, Doc, particularly in the part where where the reaction took place. So in this example, it happened in the endoplasmic reticulum, Doc.
It will happen. That is another regulations. Okay? Sequestered enzyme will be sequestered in subcellular organelle, in particular the endoplasmic reticulum. And then another one,
When enzyme binds to a substrate, forming the enzyme substrate complex, Doc.
okay, where the enzyme binds into the substrate, and then what will be formed?
An enzyme substrate complex, Doc, and it will be able to produce products later on.
A product later on. Okay, then the next regulations will be what?
Binding to other ligands, Doc.
Other ligands?
This one. Binding
Other
to the
ligands. Where will it bind?
from other ligands such as what do you call this? Magnesium, calcium, such as like that, Doc.
When there is binding on the allosteric site.
Yes, Doc. It could either activate or inhibit the enzymatic activity, Doc.
Okay. So that is another regulations. Okay? The binding of the ligand or effector into the allosteric site of the enzyme,
Yes, Doc.
of the allosteric site of the enzyme. And then the next one,
Is phosphorylation and dephosphorylation, Doc, of an enzyme.
okay, with the enzyme is phosphorylated or it is dephosphorylated.
An
Okay?
addition or removal of a phosphate group through the action of a kinase enzyme or a phosphatase enzyme, Doc.
Okay. And from here, enzyme will be either activated or inactivated.
Yes po.
Phosphorylation and dephosphorylation will activate or deactivate the enzyme. And then last will be the
Combination with the regulatory which would inactivate the enzyme, Doc.
okay. So there are regulatory proteins will be bind into the enzyme, which will result in the inactivation or deactivation of the enzyme. So these are all the regulations that could happen or could be made on the enzymes. Okay? With the enzyme. And then after that, we go to specific regulations of the enzymes. Okay? Like for example, how? How? Okay, you could go now to the next slide.
This one, Doc. This is phosphorylation and dephosphorylation.
Phosphorylation and dephosphorylation. So what will happen?
So the hydroxyl group, Doc, particularly is serine, threonine, and tyrosine, will be undergo either phosphorylation with the action of protein kinase to gain a phosphate group, or spontaneously dephosphorylated or catalyzed by an enzyme protein phosphatase, which would cleave off the phosphate group from the phosphorylated hydroxyl group, Doc.
If there will be dehydration, what will happen?
Dephosphorylation, Doc.
There will
The
be,
phosphate group would cleave off the hydroxyl group.
in other words, there is what? Removal
Removal.
of phosphate group.
Yes po.
And that could be done by what enzyme?
The phosphoprotein phosphatase, Doc.
Okay. That is when serine, threonine, and tyrosine will be dephosphorylated. Okay? And then water will be introduced and the phosphate group will be removed, will be released to form the serine, threonine, tyrosine, plus now there is now the presence of hydroxyl group on the protein substrate. And when the serine, threonine, tyrosine will be phosphorylated, and that will be in the action, presence of ATP through the action of protein kinase, serine, threonine, and tyrosine will be phosphorylated. So, as you can see, the action, phosphorylation, and then dephosphorylation. When phosphorylation occurs, it will be done by the action of protein kinase. If there is dephosphorylation, it will be done by phosphoprotein phosphatase. Phosphatase. Okay?
Yes, Doc.
Then what is your next slide?
This one shows the curve of the reaction of ATP concentration and the initial velocity, Doc, or the rate of reaction in relation to ATP concentration, substrate
Okay.
concentration.
So if there is an ATP,
Particularly on this example, I have used the substrate ATP, but this can also be seen in other substrates. But for specifically on this example, for ATP concentration, if the rate of when there is low concentrations of substrate, the rate of the reaction is slow, but it's going up or becoming high. But when the concentration of the substrate becomes high, we can see here that there is a slow progression or what do you call this? A stationary phase or a flat a
leveling
plateau
off, there is a leveling of the,
in the reaction though.
so what will happen? The maximum velocity,
It is already attained and it will just continue as is though.
okay, even if you are going to increase the concentration of
Even you add
your
the
ATP,
it has already reached the
okay,
maximum.
the maximum velocity, and there will be no change
There will be no more
in
change.
your, the maximum velocity will just merely level up or will become stationary, even if you are going to increase the ATP concentration. Huh?
Yes, Doc.
You see, okay? You increase it, your ATP from 10 to 40, what will happen to your maximum velocity?
There is a little change, however, it's not as significant as the rate of reaction from 0 to 5, though.
Okay, from 0 to 5. Or from 5 to 40.
Something like that, though.
Okay. The next slide. Okay, how gluconeogenesis and glycolysis is regulated?
Through ATP, though.
Okay, you show me the slide. Carbohydrate regulation.
Particularly, Doc, here in carbohydrate regulation, this particular pathways, I have included glycolysis and gluconeogenesis as to compare or as a example of regulation
How it's being regulated?
by the enzymes, Doc, that are present in the reaction.
That means that how these enzymes are regulated.
Um,
Can you expound?
and here, Doc, as I have mentioned,
Yeah, you have shown us how regulation of these enzymes occur. You have shown to us about the slide how this enzyme, the enlarged one.
this one, Doc. The different,
Okay,
for example, this one.
this one, that one, yes. In
So,
glycolysis.
so in here,
How this phosphoproctokinase 1 is regulated
actually, this is the
in the
one, Doc.
conversion of fructose 6 phosphate to fructose 1,6 phosphate,
If we
fructose
are
1,6 biphosphate.
Actually, this is the second one, but if we are going to go back to the first one, it's just simply the conversion of glucose to glucose 6-phosphate. However,
Yes.
hexokinase have isozymes, which are similar structures, but they have different functions. So this particular hexokinase 1, which is the most predominant one, which is present in all tissues, is being inhibited by glucose 6-phosphate, which causes a negative effect on the or negative feedback to the glycolysis. So it will not proceed to glycolysis if the hexokinase 1 will be utilized as the enzyme to convert glucose to glucose 6-phosphate. Instead, we use the hexokinase 4 isozyme, which is expressed in the liver and not inhibited by glucose 6-phosphate, making it an ideal enzyme in the conversion of glucose to glucose 6-phosphate, though.
You show me your other slide. The
Um,
one that one.
this one for the phosphofructonase. So if we're going to go back on the glycolytic pathway, we can see here that fructose 6-phosphate and fructose 1,6-bisphosphate is the commitment point of glycolytic pathway, whereas if fructose 6-phosphate will be converted into fructose 1,6-bisphosphate, it will then continue up to the last step of the glycolysis, though. So in
No,
here,
how it is? Yeah, that
in,
one.
in the
Yun
actual
is hindi
phosphofructokinase.
pinapakita dyan. It is not being shown how fructose 6-phosphate is regulated. But this one, what happened now?
Okay.
Okay, fructose 6-phosphate plus ATP. Now,
So,
how it is regulated by,
so in
and
here,
then,
by phosphofructokinase 1.
okay, how is it regulated in the presence of ATP, in the
In the
presence of citrate?
fructose fructokinase would be inhibited. Thus, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate would not be proceeded po, would not end. But with AMP and ADP, this would activate the phosphofructokinase 1, thus enabling fructose 6-phosphate to be converted into fructose 1,6-bisphosphate.
So in the presence of ATP,
Would be activated.
phosphoproctokinase is inhibited.
Yes, though.
In the presence of ATP and in the presence of citrate.
Yes, though.
In the presence of citrate.
It will be inhibited.
Why? Why it will be inhibited?
Because doc, I think because if our body recognizes that there is still too much energy in our body, there is no need for further glycolysis for production of energy. It would be just an excess and be converted into fats, though.
How about citrate?
Citrate is also a product of the citric acid cycle, which also later on produces energy or ATP. So it also inhibits the um, glycolytic pathway, though.
No, I could not understand you. The mechanism involved.
So on this part, doc, again po, when there is an excess ATP levels in our body or an excess or citrate products in our body, um, our body could recognize the energy that comes from with this. Thus, not needing not needing our body does not need further glycolysis, doc, for another ATP production, doc, or energy production, because we already have an excess.
No, but how it inhibits phosphoproctokinase so that fructose 6 phosphate will not be converted to fructose 1,6 biphosphate.
For a moment po.
You look at this other slide, letter A. What happened?
Or this one, doc.
Yes.
So in here, we can see here the conversion of the other fructose 6 phosphate that did not undergo glycolysis. It is being converted into a metabolic regulator, which is fructose 2,6 bisphosphate, doc, which now activates phosphofructokinase, thus enabling the conversion of fructose 6 phosphate into fructose 1,6 bisphosphate.
And then,
so with the activation or conversion of fructose 1,6 bisphosphate, this will now ensure the commitment of the glycolytic pathway, though, until we reach the end product of glycolysis, which is pyruvate.
so that is the case now. So if there is you explain this on the second, this one, below letter A.
This one, doc.
Fructose 6 phosphate.
Wait po.
Fructose 6 phosphate is converted to fructose 1,6 biphosphate in the presence of citrate or in the presence of
F26BP, doc.
fructose 2,6
phosphate.
2,6 biphosphate and fructose. What is this enzyme?
This one, doc.
Yes, this one. Fructose.
Fructokinase 1, doc.
Fructokinase 1. I think this is more clear. Okay?
Yes po.
It is clear. Fructose 6 phosphate is converted to fructose 1,6 biphosphate in the presence of fructose 2,6
in the presence of fructose 2,6 bisphosphate and the action of phosphofructokinase 1, doc.
and this one now in the presence of citrate, in the presence of citrate,
will inhibit glycolysis, though.
in the presence of citrate now,
once inhibited po, the fructose bisphosphatase would convert fructose 1,6-bisphosphate back to fructose 6-phosphate, thus proceeding to gluconeogenesis though. This one is gluconeogenesis. It was just cut, but it is gluconeogenesis.
Fructose 6-phosphate plus ATP will form fructose 1,6-biphosphate plus ADP. Okay, so there are two now, plus ATP. Now, if there is higher concentration in the presence of ATP, and citrate, in the presence of ATP and citrate now, this will prevent the formation of fructose 1,6-biphosphate.
Yes, Doc.
This will prevent the presence of fructose 1,6-biphosphate in the presence of ATP. And what is the primary reason for this one? Why it will prevent the presence of or the synthesis of fructose 1,6-biphosphate? In the presence of ATP and citrate?
That would lead to activation of ano, Doc, the fructose bisphosphatase 1, which would then convert fructose 1,6-bisphosphate to fructose 6-phosphate, thus inhibiting glycolysis and initiating gluconeogenesis, Doc.
Okay. Well, what I want to know, yes, that in the presence of ATP and citrate, in the presence of ATP and citrate, fructose 1,6-phosphate, fructose 1,6-biphosphate will not be synthesized. Huh? This below, fructose 6-phosphate in the presence of this again below, fructose 6-phosphate in the presence of fructose 2,6-biphosphate, in the presence of fructose 2,6-biphosphate will be able to synthesize fructose 1,6-biphosphate. Can you see that?
Yes, Doc.
Okay. So this fructose 2,6-biphosphate will is this an activator?
Yes,
This
Doc.
will activate the synthesis of fructose 1,6-biphosphate.
Yes, Doc.
This will activate the enzyme phosphofructokinase. Huh? And
Yes,
then
Doc.
in
Fructokinase.
another one is the citrate. The citrate. This will this will prevent the formation of fructose 6 phosphate, of converting fructose 1,6 biphosphate to fructose 6 phosphate in the presence of citrate.
Yes, Doc.
Huh? And this is now gluconeogenesis.
Yes, Doc.
Okay. So citrate could regulate.
Yes, Doc. Citrate and ATP would inhibit.
Okay. So that is the regulation of glycolysis and gluconeogenesis. This is only one. Okay. This is only one. Fructose 6 biphosphate, fructose 2,6 biphosphate could activate the synthesis of fructose 1,6 biphosphate. Okay, this is one form of regulation of glycolysis and gluconeogenesis. What else, Oscar?
This one is somehow related to that, Doc, because in
Okay,
it there is an involvement of hormones
okay. So hormones, insulin.
that would, for example, in this on this particular reaction, Doc, insulin, which is being secreted when the blood glucose levels are high, would activate phosphoprotein phosphatase, thus activating phosphofructokinase 2, in which we can see from the previous diagram that phosphofructokinase 2 is needed for the conversion of fructose 6 bisphosphate to fructose 2,6 bisphosphate, which is again important in the conversion of this to this.
Oh, okay. Okay.
On
So,
the other hand, we have glucagon, which is a hormone that is released when the glucose levels are depleted. So, with also the action of the cyclic AMP-dependent protein kinase, this will now activate the uh, fructo fructose bisphosphatase, which is an enzyme that would, if we can see here, this would inactivate, I rather, will dephosphorylate, rather, dephosphorylate fructose 6 bisphosphate to fructose 6 phosphate, thus not proceeding to the conversion po.
of fructose 1,6 phosphate.
Yes, and inhibiting glycolysis, thus stimulating gluconeogenesis.
And then stimulating gluconeogenesis. Inhibit glucagon. Okay, Oscar. So these are how gluconeogenesis and glycolysis is regulated. Is regulated, and of course, this by the action of insulin, the hormone, the action of the hormone. Okay, insulin or glucagon. Okay.
Yes.
Okay, who is the next reporter? Oscar? Okay.
I think it's Ma'am Jamela, Doc, or Ma'am Nori.
Ma'am?
Jamela or Miss Nori po.
No, Nori. Nori, are you present? Jim is absent. Jim Taladay. Have you reported Jim? Are you present, Jim Taladay?
Yes, Doc.
Have you reported last time?
Yes, Doc.
Next reporter would be Miss Nori Podo.
Nori, are you present? Nori,
Naka-out pa lang po siya, Dok.
out?
Opo, from duty po siya.
Naka-out siya.
Opo, pa-out pa lang.
Have you seen the second reporter? Yeah, the second reports, your second reports.
Yes, Doc. It was already posted by Ma'am Jamila po on our group chat.
So after Nori, who will be the next reporter?
Supposedly, Doc, it is Ma'am Jamila, but absent as well.
No, how about Pax? Pax is Pax not around? Absent? Is Pax absent? What
I think
happened to Pax?
Yes, Doc.
What happened to Pax? Mayroon pa siyang report. He has to make a report. So, Jem should report, Nori should report. After Nori, who is the next reporter? Supposedly, last time, who is assigned? Last time.
Ma'am Nekaida po, next to Ma'am Jen.
Michaela? What is your report, Michaela?
It's abnormal chromosomes already, Doc.
Are you ready to report that, Michaela?
Okay po.
Ah? Oh, okay. Okay, Michaela.
Okay, so good evening, everyone. I'm Micaela Alabado, and I will report about abnormal chromosomes. So, abnormalities usually occur when there is error in cell division following meiosis or mitosis. These abnormalities usually occur. These abnormalities are organized into two basic groups, the numerical and structural abnormalities. So under structural abnormalities, we have deletion, duplication, inversion, insertion, and translocation. And under the numerical abnormalities, we have aneuploidy and polyploidy. So each will be discussed later in the presentation. So before we proceed, I'm going to recap some terms and processes for your reference for this presentation. So in this slide, what's shown is the karyotype, which refers to the full set of chromosomes from an individual, which can be compared to a normal karyotype for the species via genetic testing. So shown in this slide is the normal karyotype, in which this is used to compare with other karyotypes to identify if there are any differences from it, so they can identify if there are abnormalities that I have mentioned a while ago is present in the genetic makeup of the person. Ay, sorry. So next, is mitosis. So this is the process of cell division in which the nucleus is divided into two new nuclei. This process occurs in our somatic or body cells. So our somatic cells contains both sets of homologous chromosomes with one from each of our parent. So somatic daughter cells are considered diploid with symbol of 2N to refer to two chromosomes at each pair. So these slides show the four phases of mitosis. So the first one is the prophase. This is where the chromosomes become visible when they condense into sister chromatids. So the centrioles in animal cells would move to the opposite ends of the cell. And then spindle fibers grow from the centrioles and radiate towards the center of the cell, and the nuclear envelope breaks down and nucleolus disappears. And then next is the metaphase, where spindle fibers attach to a protein structure on the centromere of each chromosome. So chromosome will line up in the middle or center or the equator of the cell, moved by the spindle fiber. And then in the anaphase, the spindle fibers attached to the centromere will pull the sister chromatids apart. So the spindle fibers will shorten, and this would ensure that each cell has the same number of chromosomes in each cell. So the chromosomes will move toward opposite ends of it, as shown here. And then after anaphase, we have telophase, where the nuclear membrane forms at each end of the cell around the chromosomes. The nucleolus will reform, and the chromosomes will become less tightly coiled and appear as chromatin again. So this would signify the completion of the nuclear division. And then it's mentioned here that the last phase is with cytokinesis. So cytokinesis is the division of the cytoplasm. So the cytoplasm of the cell and its organelle would separate into two new identical daughter cells, as shown here. So that's the end of mitosis. And it would form two identical diploid daughter cells. So next we have meiosis. So this is the cell division that happens in our gametes or sex cells, which are the egg cell for the female and sperm cell for the male. So each gamete are haploid, represented by one N or just N, which means it has one set of chromosomes. Um, the egg and sperms have only one chromosome each, which will then come together to form a zygote during fertilization. So where one chromosome from each parent in all cell will be inherited to make them diploid. So the meiosis nuclear division creates four haploid cells from one diploid cell. It's because it undergoes two rounds of division, which are meiosis 1 and meiosis 2. So as you can see from the slide, the process of meiosis is longer than that of mitosis. This is since the cell would undergo um, two rounds of cell division. So in the first round of division, the same phases are actually seen from mitosis. However, there are just slight differences from how the process occurs here in meiosis. So first, in prophase, um, it's a bit similar with that of mitosis. However, in meiosis, there is crossing over or exchange of genetic material between the non-sister chromatids of homologous chromosomes. So that's the difference between mitosis and meiosis 1. And this phenomena actually results into the genetic variation. And then next we have metaphase. So this is where the homologous chromosomes lines up at the equator. So this is actually the same as that of mitosis. And then in anaphase 1, the homologous chromosomes separate from each other. And in this phase, the sister chromatids are still together. So this is another difference from mitosis because in mitosis, the sister chromatids are separated. While in here, it's just the homologous chromosome separated, but the sister chromatids are still together. And then in telophase, this is where the cell goes through cytokinesis. So the end result would be two cells that would have unique combination of 23 duplicated chromosomes coming from both parents. So after this, the cells would undergo a second round of cell division with the same steps as the first division. The differences is that in the second division, there would be no crossovers and there will already be a separation of the chromatids. So due to this, the chromosome number is reduced by half and four haploid cells are formed, which are genetically different from each other. So haploid cells means there's only half number of chromosomes. And then these haploid cells from meiosis would undergo gametogenesis to be changed into gametes before fertilization can occur. So that's all for the recap about cell division. So now we will proceed with the chromosome abnormalities. So as I have mentioned, there are two groups, numerical and structural. So we will discuss first about the numerical abnormalities where aneuploidy and polyploidy are classified under. So, aneuploidy is an abnormal number of chromosomes that occur when an individual is missing either a chromosome from a pair, which would form monosomy, or has more than two chromosomes of a pair, which forms trisomy and even tetrasomy. So in humans, an example of the condition caused by by a numerical anomaly is Down syndrome, also known as trisomy 21. So this is an individual that has three copies of chromosome 21 rather than two, as shown in the slide. Okay. So it is said that trisomy has been determined to be a function of maternal age. So the older a woman gets pregnant, the higher the risk of having a child with trisomy. And then for an example of the monosomy, since I've mentioned monosomy is also under aneuploidy, we have Turner syndrome, where the individual is born with only one sex chromosome, which is an X only. So it's also shown here in the slide the karyotype of an individual with Turner syndrome. So it's in its sex chromosome, it only has one, which is only X. So next numerical abnormality is polyploidy, in which a normally diploid cell or organism would acquire one or more additional sets of chromosomes. So in other words, the polyploid cell or organism has three or more times the haploid chromosome number. So it's the whole set of chromosomes that is multiplied here in polyploidy. So polyploids are actually common among plants as well as among certain groups of fishes and amphibians, but when it happens to humans, it's lethal. So polyploid in humans can form, humans come in form of triploidy and tetraploidy. So triploidy occurs in about 2 to 3% of all human pregnancies and around 15% of miscarriages. The vast majority of triploid conceptions end as miscarriage. And those that do survive to term typically die shortly after birth. So in some cases, survival past birth may occur longer if there is mixoploidy with both a diploid and a triploid cell population present. So complete tetraploidy is actually a more rarely but diagnosed condition than triploidy. But this is observed in 1 to 2% of early miscarriages. So just to differentiate, aneuploidy is more common in humans, commonly observed in humans than polyploidy, since polyploidy is a lethal condition in human beings. So for the cause of the numerical abnormalities, disorders of chromosome numbers are caused by non-disjunction. So this occurs when a pair of homologous chromosomes or sister chromatids um, fail to separate during meiosis 1 or 2, or even during mitosis. So shown on the slide is a diagram from meiosis 1 where non-disjunction took place uh, when this junction took place in meiosis 1 and resulted to um, two of the daughter cells having the two, which resulted to two of the daughter cells um, that would have possibility of forming trisomy in the future, and then the other two daughter cells have um, has a chance of developing monosomy. And then in this slide, it shows the non-disjunction that happened in meiosis 2. So with the sister chromatids, instead of the homologous chromosomes failing to separate. So again, some of the gametes contain extra. So we have one daughter cell with an extra chromosome and one daughter cell with missing chromosome. So when an aneuploid sperm or egg combines with a normal sperm or egg in fertilization, it makes a zygote also aneuploid. So for instance, if a sperm cell with one extra chromosome would combine with a normal egg cell, the resulting zygote or one-celled embryo will have a chromosome number of 2n plus 1 since since the sperm has additional chromosomes in it. So next would be the structural abnormalities where chromosome structures is altered and would take several forms. So we have five types of structural abnormalities. So first is deletion. It is where a portion of a chromosome is missing or deleted. So known disorders in humans include Wolff-Hirschhorn syndrome, which is caused by partial deletion of the short arm of chromosome 4. And then we also have Jacobsen syndrome, also called terminal 11q deletion disorder. So, um, as shown on the slide, the original form, in the original form, a part of it was deleted. And then this is the one that is manifested into the person. So because of the deletion of this segment, depending on which chromosome it happened, it can manifest into Wolff-Hirschhorn syndrome or Jacobsen syndrome. So next is duplication. So a portion of chromosome is duplicated. So it results into extra genetic material. So known human disorders for this kind of abnormality is the Charcot Marie Tooth disease type 1A, which is caused by the duplication of the gene encoding peripheral myelin protein 22 on chromosome 17. So as shown in the diagram, this portion of the chromosome got duplicated. And then next is inversion, where a portion of chromosome has broken off, turned upside down, and reattached. Therefore, the genetic material is inverted and the sequence is disturbed. So, even if there is no deletion of the sequence or even duplication of the sequence was inverted. So, since the chromosome is very particular, when it was it's very particular with the sequence, as it's basis in the what would be manifested in a person, disturbance of this would also cause problems in uh, the manifestation of the traits to the person where this type of abnormality happens. So there are actually two types of inversion. So it can be pericentric or paracentric. Pericentric is the type of inversion which includes the centromere, while the paracentric inversions do not. So the pericentric inversion can change the relative length of the chromosome arms, while the paracentric inversion cannot. So an abnormality caused by pericentric inversion in humans is the chromosome 9 inversion, which is one of the most common structural balance chromosomal variants, with an estimated incidence of about 3.5%. So it actually remains unclear if this rearrangement have big clinical significance, but in some cases, it has been associated with congenital anomalies, growth retardation, infertility, recurrent pregnancy loss, and even cancer. So next we have insertion. This is an abnormality where a portion of one chromosome has been deleted from its normal place and is inserted into another chromosome. So an insertion can be direct or near-ins if the segment keeps its orientation in relation to the centromere. So like this. So this is the original and this is the direct insertion. And then an insertion can be inverted if the most proximal band becomes the farthest from the centromere, the distal band number preceding the proximal one. So this is the an example of the inverted insertion. So as shown here, the proximal band becomes the farthest from the centromere. So there's also inversion in the portion of uh, there's also inversion in the portion that is inserted to the other chromosome. And then we have translocation. This is where a portion of one chromosome is transferred to another chromosome. So there are two main types of translocation. We have the reciprocal and the Robertsonian. So in the reciprocal translocation, segments from two different chromosomes have been exchanged. While in the Robertsonian translocation, an entire chromosome has attached to another at the centromere. So in humans, they only occur with chromosomes 13, 14, 15, 21, and 22. This is because these are the what we call acrocentric chromosomes. So they have their centromere situated to one chromosomal arm that is much shorter than the other. So this type of abnormality occurs only in these chromosomes that are considered acrocentric. So Robertsonian translocation is a common form of chromosomal rearrangement that happens in humans. So during the Robertsonian translocation, the participating chromosomes break at their centromeres and the long arms fuse to form a single chromosome with a single centromere. So the short arms also join to form a reciprocal product which typically contains non-essential genes and is usually lost within a few cell divisions. So for the causes of the structural abnormalities, um, there are actually four that I have read that may have caused the structural abnormalities. So we have errors in the chromosomal crossover, which I mentioned happens in the meiosis 1. And then it can be caused by chromosome breakage, inheritance, and DNA mutation. So for the chromosomal crossover, so as shown in this slide, and as also mentioned a while ago, crossing over is the swapping of genetic materials that occurs in the germline. So if there is any error that would happen in between this process, it may result in any of the abnormalities mentioned. It can be deletion of the genetic material, even the duplication of it, insertion of it to other chromosomes. So that could happen if there is any error in the crossing over um, process of meiosis. And then next is chromosomal breakage. So most of the time, just chromosomes can break and heal correctly. However, problem would arise when they do not heal correctly and instead are deleted or rearranged. And then one is inheritance. So there are chromosome abnormalities that can be inherited from a parent. So as shown in the slide, if one of the parents is actually affected with an abnormality, there is a chance that their child or offspring can also get the abnormality. And then next is DNA mutation because changes in the DNA sequence can also occur at the level of chromosome. So in which large segments of chromosomes are altered. So in this case, fragments of chromosomes can be deleted, duplicated, inverted, translocated to different chromosomes or otherwise rearranged, which would result to modification of the gene dosage. And then complete absence of the genes or alteration of the gene sequence. So for further explanation on the different kinds of mutation, it will actually be discussed by the next reporter, which is Mr. Alex Cruz. So that's actually the end of my report for abnormal chromosomes. Thank you.
Okay, Michaela, I think we will end up in here and then we will continue our discussion next meeting.
Yes po.
How about that?
Is there any, is everything explained, Doc, for abnormal chromosome?
Okay, can you do again? I think we will discuss this next meeting about
Okay.
your report. Huh?
Okay, Doc. But there
Okay?
is no lacking information that I should
Okay.
keep.
Okay. No problem.
Okay,
Okay,
Doc.
see you again next meeting. Okay, and please prepare your reports, the second report.
Okay, Doc.
Okay?
Thank you.
Okay, thank you.
Bye, guys.
Nice one,
Bye.
Mikey.
Bye, 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 bye.
Bye.